Hi everyone, this is Mr. Hall and this is a video about the Enzyme Required Practical that you will have to do for your biology course. This is like an essential experiment you have to know for any biology course sort of pre-16. So if you're doing AQA, high school chem, whatever it is, you do have to know the key details of the experiment. So because it's so important, I think it takes more than one video to really go through it in the kind of detail that you need. So I am going to do this video about the method and the kind of questions that you might be asked about the method. And then in a future video, I'll talk about data and analysis and how you deal with all that kind of stuff. So today, I'll just be talking about the description of the procedure and to some extent the safety measures for an investigation into enzymes and pH. Now, pH is not the only thing I'll be talking about. I'll also talk about if you want to change temperature as well. It's just sort of a flipping of the different variables that you could encounter in this experiment. So first off, the most common experiment you'll do is where you break down starch, which is a carbohydrate, using the enzyme amylase, which is a type of carbohydrase. Okay, and iodine is a really good indicator in this experiment because when iodine turns blue or black, starch is present. But if it stays orange-brown or turns orange-brown, there's no starch. So if you look at this picture over here, this is an indicator where the iodine is orange, so there's no starch. Whereas over here it's deep black, so there's lots of starch. Okay, so the process that basically goes on here is here's your carbohydrate starch, which is basically a long chain of different sugars bonded together. And then what amylase does, or carbohydrase, is that it breaks it down into smaller pieces. Okay, like so. So this is your starch, and this is your basically your glucose, which is a type of sugar. All right. Now, because there's no starch here, if you were to put iodine on it, it would look orange. However, here, this starch, if you were to put iodine on it, it would turn blue-black, okay? And that's basically the indicator to show whether your starch has been broken down or not. It should initially look very dark, and then over time, it will start turning orange, okay? So, I'm just going to go through the key steps of what you would do in this experiment. So the first thing you would do is you'd measure 10 centimeter cubed of starch solution. Now, starch solution, it's a bit of an inaccurate term because starch isn't very soluble. So what you'll have is sort of a little bit of liquid with a lot of sort of particles floating around it, a lot of precipitation, if you like. And that's because the starch isn't very soluble. So it will be a liquid and there'll be sort of bits and bobs floating around in it. And you put it in a boiling tube because you'll be warming it later on. The second step is that you add a buffer solution into this. Now, a buffer solution is basically a liquid which determines the pH of it. Now, let's say that you are wanting to change the pH or do this experiment at different pHs to see how pH affects enzyme activity. So you might have a buffer solution at a pH of 1 or 2, 3, 4, 5. It could be any, right? And in most experiments in high school, you might have a buffer solution at 7, one at maybe 3, and one at 11 or 10 or something. Now, for the sake of argument, let's say that we use the pH solution, a buffer of 7. Now, you don't need much of it. You just need like 1 centimeter cubed. So we'll put it inside a pipette and we'll just drop 1 centimeter cubed inside, and then we'll mix it all around. So then we have starch at a pH of 7 because it's got our buffer. Now, the next thing we do is we measure 1 centimeter cubed of amylase solution using a plastic syringe, and then we add it to the test tube. Now, remember, we've already got our starch solution at a pH of 7 because of the buffer. This one is what's going to contain the amylase. The important thing is that it's in a different test tube. Okay, so again, we can do it using a pipette. We just drop 1 centimeter cubed inside. You don't need a lot of enzyme for this experiment. And then what you do is you put both of those boiling tubes or test tubes inside a beaker of water to warm up. Now, the ideal temperature is 37 degrees. Reason for that is because that's the temperature of the body, and that's the temperature that most enzymes work best at. Okay, if they're not at that temperature, they don't work particularly well. So you want to control that temperature. Now, putting it in a beaker like this is great, but it does have its problems because heat can escape and cool down. So therefore, your enzymes might not be at the optimum or perfect temperature to work. Therefore, using a water bath like this is very, very effective, okay, this water bath, because it controls the temperature. So it will always sort of warm it up a little bit and keep it at 37 degrees. It's like basically like a little shallow bath where you can put test tubes in and it's got a filament or something inside it to keep it warm. It's really, really good for a scientist. Okay, so those test tubes are inside there and they're warming up. In the meantime, you could put a drop of iodine solution inside each well of your spotting tile. Now, a spotting tile is like a rectangular piece of plastic with depressions inside it. and you can see that I've put a drop of iodine, well, in this picture there is a drop of iodine in each well. Okay, those depressions are called wells. And what you're then going to do, once you've done that, is you're going to add your amylase to your starch solution. So this is my starch at the pH of 7. This is my amylase. I add one inside the other. So let's say, for example, I add my amylase inside here, and therefore what I'll end up with is a solution that contains starch and amylase. Now this is important because this is the first time at which the amylase is going to start breaking down the starch. Okay, so you've got your starch molecule that looks like a massive chain of sugars sort of bunched up together. You've then added your amylase, which are these pair of enzyme scissors, which are going to start trying to cut up that starch into smaller pieces. We just want to see how effective and how quickly you'll do it in different pH conditions. So that's happened, and then what you're going to do is you're going to take a drop of that starch amylase mixture and add it to a well in the spotting tile. Now this is quite important. Let's consider this as being 0 seconds. So as soon as it's added, give it a bit of a mix, and then you're going to put one drop inside here. All right. Now at the very start, what you might find is as soon as you add it, I'm going to use a different color here, you might find that it goes really dark blue-black, like that. Now that kind of makes sense because starch turns blue-black in iodine. Okay, so that's exactly what we'd kind of expect, and that's fine. You then do the same thing after 30 seconds. So after 30 seconds, we'll get another drop and we'll put it inside the next spot. And again, what you might find is it will turn dark again. And the reason why that's happening is because it's going to take some time for all of that iodine to break down. Okay, because if you remember, amylase breaks down starch, but it needs time to do that. So this massive chain here of the molecule starch, it might take something like 3 minutes for all of that to be broken down by the amylase. So what you might find is as the minutes progress and you keep doing this method, it might take, so this will be like the 90-second mark, the 2-minute mark, and this will be 180 seconds, and this will be 60, 90, 120, and this is 180, so this is a 3-minute mark. So what you might find is over time it becomes slightly less blue and slightly more orange until it turns perfectly orange at around 210 minutes. And what that tells you is that it takes 3.5 minutes for your starch to be broken down by amylase at pH 7. Now, if you do it at different pHs, what you might find is it takes longer or shorter amounts of time to break it down. And those are basically the next steps. So for step 9, it's to repeat it every 30 seconds until there's no color change present. So basically it goes from black all the way down to orange and stays orange, all right? Like so. And then you repeat steps 1 to 9 for the same pH value. Now, the reason why you want to do that is to make sure that your data is repeatable. And what that means is that you do either the same or a very similar experiment more than once and you get very, very similar results, or you come to the same conclusion. So if I did this a second time and then I found that after, sort of stays black, and then after about 3 minutes, which would be at sort of around this point, it then goes orange. If I get the same result, that's a good sign that it's repeatable. All right, well, can't really see that. And then the last thing you do is that you repeat all those steps, but then you would use a different pH buffer. Now, if you remember at the start, I used the example of doing this at pH of 7. After doing it twice, to get 2 sets of results, you then might want to do it at pH of 3 and a pH of 10. And then what you do is you get a really good idea as to which pH this particular enzyme works best at. Okay, now in terms of planning and exam questions, there's basically 3 areas. After looking at all the questions available, there are 3 types of questions you might get. The first type of question is asking for variables. So I'll ask you what's the independent, the dependent, and the control variable. So if I just show you this example of an investigation, it says, in an investigation, some pupils set up 5 test tubes. Each tube contained 1 centimetre cube of protein and 10 centimetre cube of protease solution. Each tube was kept at a different temperature for 24 hours. And they might say give 1 control variable. Now remember, a control variable is the variable that you want to keep the same, and there's always more than one. And by looking at this method, you can see there's loads of stuff they're keeping the same. They're keeping the amount of protein the same, they're keeping the amount of enzyme the same. So you'd say, and be careful here, don't say amount because that's a bit sloppy. I would say here the volume of protein, or you could say the volume of enzyme, or, and this is kind of not really mentioned here, but you would have to keep this the same, the pH would have to be kept the same as well. So you should always in any investigation be able to pick up at least 3. Never mention equipment, all right? Equipment is poor because in most cases it will have a negligible effect on results. Another question here is, what was the independent variable? Well, that's pretty obvious from what they're saying here. The independent variable is what you're going to change, okay? And in this investigation, it's the temperature. Now, basically in this investigation, there are 2 things that are going to change. It's either going to be the temperature or it's going to be the pH, all right? And the other one that you're not changing automatically becomes your control variable. So just be aware that asking for variables is a very popular one to ask for. Second type of question is answer for a hypothesis with scientific reasoning, all right? So I've got the same investigation as I mentioned before, except I've specified the temperature. And the question you might be asked is which temperature would show the greatest enzyme activity? Explain your answer. Okay, now they might reword this in loads of different ways. So they might say, which temperature would show the most digestion in the least amount of time, or something like complicated. But essentially it boils down to which one's going to work best. And you hopefully will know that the temperature that works best out of these 5 is 40. Okay, so you'd say 40, and then the reason for that is because this is a temperature at which enzyme work best, or the optimum temperature. So a temperature which enzymes work best. And the best word for like the temperature or the pH in which they work best is optimum. So it's optimum temperature, optimum pH. All right. Now the final type of question you might be asked is for, sorry, don't know why I've repeated that, is for a 6-mark method. Now this comes up sometimes. I haven't actually seen many cases of this with enzyme activity, but what they might ask you is they'll just give you loads of lines. They'll say like plan investigation to see how the activity of amylase is affected by pH if added to starch solution. And you'd basically have to replicate that method, which I described before as a 6-marker. It's quite a long thing to do, but quite straightforward if you remember the steps. What they might also ask you to do is improve an experiment. So they might give you like basically a terrible experiment of like 2 lines and say like improve it. And if they do that, it often means making the method valid. And when you're doing this, there's basically a couple of things you always have to do. Always brainstorm your keywords and always brainstorm your variables. So for example, always think about what is my independent variable, what is my dependent variable, and crucially, what are my control variables? Now, control variables is probably the most important one because it harks directly back to the idea of making it valid. So think about what volumes of solutions you need. Think about how you're going to control pH, how you're going to control temperature. And remember with pH, if you're controlling it, it's going to be with a buffer solution. And if you're going to control temperature, it's going to be with a water bath. All right. And these are all the things you have to keep an eye on. All right. So I hope that's helpful. And in a follow-up video, I'll talk more about how you analyze the data.